Although our primary mission is to serve our Bay Area customers with reliable, high quality, and affordable water, our stewardship policy commits us to responsible natural resource management methods that protect and restore viable populations of native species and maintain the integrity of the ecosystems that support them. Our watersheds contain almost 20% of California's native flora and over 300 species of birds, reptiles, mammals, and amphibians. 35 of these plant and animal species possess endangered or special status classifications, and another 50 of our documented species are considered locally rare or unusual for our area. Since parts of the regional system were more than 100 years old and seismically vulnerable, the city launched a voter-approved $4.6 billion water system improvement program with more than 80 capital improvement projects in 2002 in order to improve reliability. 16 of those projects occur in the watersheds and disrupted habitats that had not been disturbed in over 80 years. Other projects have incredibly large footprints and occur in remote areas of the watershed. The reconstruction of Calaveras Dam, which triggered restoration efforts at several sites in our Alameda County watershed. The habitats impacts associated with these projects required a large amount of mitigation. We chose to keep the restoration projects on watershed property, and the mitigation effort restores almost 2,000 of the 60,000 acres of watershed. Our implementation of the habitat restoration projects included a comprehensive invasive species program that attempted to control for invasive aquatic, plant, insect, and pathogen species. So, although our pest and pathogen-free specifications were comprehensive, much of these specifications were new to the industry and met with skepticism at the time. And in January 2014, we started to observe dying plants in the field and confirmed our first detection of Phytophthora in a habitat restoration site. At the 2014 Science Symposium, our habitat mitigation engineer, Greg Lyman, outlined the precautions we incorporated into contract specifications for heavy equipment and imported materials for these projects, as well as our nursery inspection and enforcement program, where despite all these efforts, we ended up with 10 species of Phytophthora at 6 habitat restoration sites. In the fall of 2014, we had just installed 9,000 solarization basins to try and heat treat infected planting basins, and we altered our restoration approach and started using only seeds and on-site divisions for restoration. Since we are no longer using container plants at our site, this has greatly increased the amount of seeds that we need to collect in order to do in situ sowing. We now have to incorporate detailed seed treatment, seeding rates, and seeding methods into our specifications for the contractors who are implementing the projects, and in some cases, we are dealing with sowing super small seeds into planting basins that were set up for container plants. And in many instances, we also don't have a lot of information regarding seeding rates in the field for the taxa we're attempting to plant. Since January 2015, we have now taken an additional 383 samples in an effort to not only target individual infections in our plants, but we have also been attempting to quantify spread of the pathogens from known infected basins. Our small amount of spread sampling that we've done at this point has looked at 4 different Phytophthora species, and we've only been able to confirm positive spread of 1 species, that's a P. cambivora, and the farthest one away was 1.5 meters. We've also confirmed 6 new plant host species that were showing signs of infection, and the biggest surprise for us with these efforts was the identification of P. tentaculata on Artemisia douglassiana, which Ted mentioned. Another element of our sampling efforts has included water sampling. We've taken 110 water samples from the edges of the watersheds to get an idea of inputs from adjoining properties, as well as numerous samples from streams within and upstream of our habitat restoration sites. 85% of our stream samples have been positive for Phytophthora, and we've found 20 of our now 30 species in those water samples. Sorry, Where's my water sampling slide? As our sampling efforts have expanded and our species list has grown, we wanted to know more about our detections, and we provided support to the UC Berkeley Forest Pathology and Mycology Lab and the Forest Service to author a literature review of the species in order to assist our planning and response efforts. They are planning on publishing this literature review, so it should be available for anybody who's interested in learning more about the ecology and biology of these selected species. We also provided funding to investigate the susceptibility of California sycamore, coast live oak, and toyons to P. tentaculata and P. cactorum. These two pathogen species we were interested in investigating to determine their potential impact on California ecosystems. The quick summary, the bullet point summary of their results was that coast live oaks were susceptible to both pathogens in their roots and stems. The toyons had small lesions, and so the researchers were recommended that they needed to be tested carefully because they could be asymptomatic carriers. And the sycamore roots were shown to be resistant, but the stems were highly susceptible, and so they recommended to avoid splash on a sycamore stem, which is something that's a little hard to do in a riparian corridor. In select hot and cool locations at our solarization sites, we've installed soil probes for monitoring to see if the solarization effort was effective. And we've been reaching lethal temperatures at up to 4 inches in some locations, but we've never reached lethal temperatures at our target depth of 8 inches. And at some of our shaded and cooler locations, we're only reaching lethal temperatures at 2 inches. Since we aren't sure that solarization is the best fit for all of our locations, we started investigating the use of steam to treat the pathogens in the field. And last spring, we purchased a steam auger from the Hansen Lab at UC Davis. The auger has to be maneuvered through the sites with either a tractor or a skid steer and connected to a boiler and a water tank in order to produce steam. And we're in the process of purchasing a boiler and evaluating our sites in terms of accessibility. We're also working with Phytosphere Research and Washington State University to create a protocol outlining the use of this equipment in the field in a habitat restoration setting. We've contributed a small amount of funding to consultant H.T. Harvey for a pilot project to determine if ecological scent dogs could provide an effective and efficient method to accurately screen for Phytophthora in either a field or nursery setting. This pilot is a 2-phase training approach that's being conducted with 4 Phytophthora species, and it's still in its early stages, but the preliminary results have been very promising. In addition to our sampling and research efforts, we have decided to construct a temporary nursery that will use the best management practices as outlined by the Phytophthora Working Group Nursery Subcommittee to grow over 30,000 landscape plants for our Sunol Long-Term Improvement Project, which is located on the edge of our watershed in Sunol. The nursery will only grow plants from seeds collected in our local area. And key BMPs we are aiming to incorporate include having good drainage and getting the plants up off the ground, decontamination measures for people entering the nursery and for pots, equipment, and vehicles, not using fungicide, heat treating our soil, and a fairly rigorous testing regime. In order to heat treat our soil, we're purchasing a steam wagon that will be used in conjunction with the boiler to treat soil in the nursery. We're also planning to use it at in situ propagation beds that we'll use at our restoration sites to amplify seeds on site. So what we know now is that our mitigation timelines are being extended because they were developed under the assumption that we would be using container plants, and the timeline extensions are greatly increasing the cost of our implementation. We know that less is better when talking about planting volumes and planting approaches, and that being clean and staying clean needs to be an organizational commitment. Once you are infected, it is very hard to treat these sites, and it's also very expensive. So with that, I will thank the many, many contributors who have been helping us and pass it on to the next speaker.